It's blueberry season. Aren't you excited? I didn't expect that many people to say woohoo, because I'm expecting you to say, hang on, that's not until July, August. And you're right. The best time for ripe fruits of the wild or low bush blueberry, Vaccinium angustifolium, is in August in Maine. But for each and every one of the millions upon millions of tiny white flower of juicy blueberries in August, there's a tiny white flower back in late May, so right about now, that's getting pollinated by an insect, most likely a bee. But not just any bee, though there are about 100 species of bees that live in Acadia National Park, only about 20 of them are known to be any good at pollinating blueberry flowers. These include the bumblebees and also a group of small bees in the genus Osmia, which are so good at pollinating blueberry flowers, they're known as the blueberry bees. So, bees pollinate flowers, and it's awesome. But what's new? It might seem pretty simple, actually, but it's not. The blueberry plants are responding to a complex set of environmental cues to decide to open their flowers exactly now. And the bees are also responding to a complex set of cues to decide to emerge from their winter hibernation also exactly now. So, what if those environmental cues start to shift and change? What if, because of climate change, we have a particularly warm year, maybe like this one, and the flowers, but not the bees, emerge early, and they go unpollinated? Or what if the bees emerge early, but it's too early for the flowers? Or what if we have a cold year, and either the bees or the flowers, but not both of them at the same time, emerge late? And that's just temperature. There's also precipitation, pollution, habitat loss, invasive species, and disease to wonder about. The answer to all of these questions is we don't know. What we do know is that because of climate change, these kinds of scenarios are going to become more and more likely over the coming years and decades. And it's not just blueberries and bees. There are around 800 vascular plant species that grow in Acadia National Park, and 5,600 species of insects. And it's not just plants and insects either. There's all the other animals, and the fungi, and the microbes, and their interactions with each other. Think about the peregrines that are just starting to nest on the precipice on the side of Champlain Mountain, near where I took this picture. Think about how they have a complex relationship with, a reliance on their prey species to survive through the year. And so, I'm concerned. I'm concerned about this place that I love, Acadia National Park. This climate change and other environmental changes that humans are causing leads to a set of big unknowns, and they're complicated. And frankly, it's scary. And one of the reasons it's scary is that, of course, it's not just Acadia, and it's not just MDI or Maine or the United States. This is a global concern. The Intergovernmental Panel on Climate Change says that its number one concern about the impacts of climate change on a global scale is how it will affect biodiversity. Biodiversity is the mind-boggling assemblage of living organisms with which we share our planet. And it's a concern because we rely on all these species for our food supply, for our medicine, for the stability of our environment, and for our sheer enjoyment. It's also a concern for science. How can we as scientists wrap our heads around how to study such a big question like how is climate change going to affect biodiversity? And when I say study, I don't just mean understand, I also mean monitor and predict and maybe even mitigate some of the more serious changes. Eminent British ecologist Georgina Mace has called for a new kind of ecology, a new kind of ecology predicated on scaling up and sharing data and collaboration. In other words, we need more data. Here's the tricky part. To gather these kinds of data, we need to go out there and we need to collect or observe individual specimens, and we need to identify them down to the species level. The problem is that identification is hard, and it's time-consuming, and there's a dwindling number of taxonomists worldwide. The United Nations Convention on Biological Diversity thinks this is such a big problem that they've given it a name: the taxonomic impediment. The taxonomic impediment. Today, I want to talk to you about what we might be able to do to help overcome the taxonomic impediment. Indeed, what you might be able to do to help overcome it. Because I think a big part of the solution is something called citizen science. Citizen science is when members of the public participate in scientific research. It's good for science because it generates a lot of data, and it's good for the participants because they have an educational experience and they get to feel like and be part of the solution. There are citizen science projects cropping up everywhere these days. Some that you might have heard of include eBird, Bat Detective, iWire, where you get to help map the human brain by playing a game online, Galaxy Zoo, and the whole host of projects that are part of what's now known as the Zooniverse. So maybe citizen science can help us overcome the taxonomic impediment. But I just told you that identifying specimens is hard. And scientists are rightfully wary of identifications made by citizen scientists. Heck, I'm a scientist, and I can barely identify things. Fortunately, there's a new crop of tools emerging to help with specimen identification. These include iNaturalist and iSpot Nature, which use crowdsourcing to help identify photos of specimens uploaded by citizen scientists. And there's LeafSnap, where you take a picture of a leaf on a white background, and then it uses the outline of the leaf compared against the outlines of leaves from known species to help identify it. These tools are all really exciting, and they have a lot of potential, but a lot of species are going to fall through the cracks, maybe literally because they're too small, or maybe because they're not popular or not particularly amenable to those tools. Today, I want to tell you about another tool for specimen identification. It's called DNA barcoding. DNA barcoding is using a specimen's own DNA to try and figure out what species it belongs to. It's sort of like the idea that many of you have probably heard of, where you use the DNA from a human sample found at, say, a crime scene and compare it back to a database of DNA samples from known criminal suspects. DNA barcoding is a lot like that, except instead of criminal suspects, our database is filled with DNA from the 1.8 million known species of animal, plant, and fungus that have been named by science. It works like this. You go out there, you observe some organism, and you take a small sample, say just a tiny hole punch from a leaf or the leg or wing of an insect. Back in the lab, we extract the DNA out of that sample. That means we separate it away from all of the other cellular debris in the sample. Now we've got all this DNA. We actually have several billion nucleotides of DNA from each and every sample. Nucleotides are the A's, T's, G's, and C's that make up DNA's four-letter alphabet. We don't need nearly that much information, though, to identify a specimen. We've found over the last decade or so that we only need about 700 nucleotides to identify a specimen to the species level. The 700 nucleotides from a gene called CO1. So what we do is we isolate and we sequence or read the CO1 gene from our unknown sample, and we compare it back to our database of CO1 sequences from all the known species. These sequences exist in a place called the Barcode of Life Data Systems, or BOLD. We literally copy and paste our 700 nucleotide sequence into the BOLD identification engine. So it's like we're Googling a really long word. And we click submit, and just a few seconds later, we get an identification. We also get a lot of information about the species that we've just identified and links away to other sources of information. It's pretty straightforward, actually. It sounds like something you might have seen on CSI. But actually, it's taken over a decade of international collaboration to decide on what gene regions to use, what our data standards should be, and to work with museums and other specimen collections to access samples for DNA sequencing. Meanwhile, we've been depositing these sequences into BOLD, and now we're up to nearly 5 million CO1 sequences and sequences from other genes in the BOLD database. So we're nearly there. Not every single species is represented there yet, but we are to the point where we can get a pretty reliable identification of a specimen based on its DNA alone. But how is this going to help us with the big, grand, global question, how does climate change affect biodiversity? Here's what it could look like. Imagine thousands, hundreds of thousands, millions of citizen scientists fanning out across the world, at their homes, at their work, in their favorite national parks, and other vacation spots. And they're gathering data, they're making observations, they're noting their location, and the date and time of their collection, and they're taking a small sample using a non-invasive technique like swabbing so they don't actually damage the environment. Then they drop their swab kit in a drop box or mail it back to a DNA barcoding laboratory where an identification is made. The identification is then sent back to the citizen scientists so they can see what they found. And it's also communicated to the researcher whose data needs were the whole point of the project in the first place. Working at the MDI Biological Laboratory, I collaborated with Acadia National Park and the Schoodic Institute to try out this idea. We invited members of the public to the sandbar between Bar Harbor and Bar Island, so literally just behind us here, to collect marine invertebrate specimens from tide pools for DNA barcoding. One of the things we found was this little shrimp. It was a surprise to our taxonomist collaborator. She had ruled it out because it's not thought to live north of Cape Cod. Maybe it's actually migrating northward in response to climate change. Because of DNA barcoding and citizen science and the combination of those two things, we know this is even a question that we can ask now. And so we're working to expand our project beyond Frenchman Bay, beyond the Acadia region, to other regions and maybe even other countries. But you don't have to wait. There's something that you can do starting right now, today, after you leave here. You can participate in a citizen science project. These links will help you find a project that might best suit you. You can also upload images and maybe even try your hand at identification on iNaturalist.org. There are bees buzzing around the blueberry flowers out there right now, just waiting for you. Thank you.